Also, on a longer time scale, you can see them disappear. So we have now collected many gigabytes of optical data, and we are in the process of analyzing this data to look at the kinetics of assembly and disassembly. But our goal is not just to look at these complexes at a level of this chromatin indicator, but also to see the repair proteins themselves that are present in single copies in living cells. So this is a goal we've not yet achieved, but we hope to achieve with our quantum dot-ready proteins that we've made and are in the process of conjugating. It is going to require advanced microscopy. Optical microscopy is ordinarily limited by diffraction to about 250 nanometers in the XY dimension. Usually it's more like 750 in the Z dimension. We are applying new instrumentation with high sensitivity cameras, noise reduction algorithms, and a technique called structured illumination, where multiple images are collecting using patterned illumination light and combined. This is just an example early in the testing of this microscope. It's a conventional microscope image of some subnuclear structures, and this is with structured illumination. You can see the much better resolution, and in this short movie that just played, you can see this resolved in 3 dimensions. So we're very hopeful about the promise of these technologies. I do want to talk, though, about a clinically relevant example of how we can modify this machine. And this has to do with modifying the response to radiation therapy. So there are something like 1 million patients a year who will receive radiation therapy in North America. It's a very mature technology. Sometimes radiation therapy is given to prevent the recurrence of a cancer. Like early stage breast cancer, it's excised and the woman receives radiation in the hope that that will reduce the risk of recurrence and death. But a lot of times radiation therapy is given for cancer control. An example would be lung cancer. Most people that have lung cancer, particularly when it's advanced to a certain stage, will receive radiation as part of their treatment. It clearly extends life, reduces some of the symptoms that are associated with the cancer, but it does not cure people. The problem is that radiation there's not enough therapeutic gain. And we just heard from Dr. Blumberg about this problem that war on cancer is a war on ourselves. There's some gain from radiation therapy. That's because tumor cells lack, they're dumb, they go on dividing in the presence of DNA damage, and they tend to, they're more susceptible to radiation injury, but not that much more susceptible. They replicate their unrepaired DNA, they divide, and they undergo post-mitotic cell death. So the idea that we had is if we could tune the double-strand break repair machine, if we could delay the process, you wouldn't have to completely inhibit it. You just have to delay it long enough for this competing process of cell division to happen in the tumor, and you would increase the therapeutic gain that's there naturally from radiation therapy. And of course, if you could target the therapy, that would improve it. If you could physically target it, it would improve it further. I think that would be true of any cancer therapy. If you could physically target it, you would improve it further. Here's the concept in cartoon form. Remember, there were 6 polypeptides that assembled. And what we have is we have an antibody fragment, that binds right in the middle of the 3rd polypeptide and prevents those phosphorylation events and prevents the further assembly of the complex, and you're left with a poisoned, incomplete repair reaction. Stephen Lee talked about single-chain antibodies yesterday, introduced them. They're 3 to 4 nanometers in size. They're produced recombinantly. We happen to be working with one that has very high affinity for its target, which has been precisely mapped in a conserved regulatory sequence in the center of this key enzyme in the repair pathway. So antibody therapy is very well accepted for cancer treatment, but all the therapies we have work on the outside of cells, and I'm proposing to use this really nice specific platform, but on the inside of cells. So how would you deliver the antibodies? Well, we proved the concept by micro-injecting them into cells, and we observed a lot of sensitization to clinically relevant radiation doses. Microinjection is nice. It may have applications to other clinical problems, although clearly not to cancer, because of the precise volume and timing of the introduction of the protein. You can use unmodified proteins. You don't need to modify these in any way with carriers, and you have control cells that are uninjected on the same plate. And my colleague, Dr. Shu-Yi Lee, talked quite a bit about these proof of concept studies last year. But today I want to talk about a more practical delivery method, which is receptor-mediated endocytosis. And this allows for cell-specific targeting. It's a fairly mature technology. It's been used for other things. And importantly, it offers a path for clinical translation. And in particular, we're using folate receptor-mediated delivery. Folate receptor is overexpressed in many cancers. Folate's a cofactor for several of the reactions of DNA synthesis, and cancers do a lot of DNA synthesis. Binding to the ligand promotes non-destructive internalization and release of cargo, proven for model proteins, and we know that it would be translatable to the clinic if we could get it to work. We coupled the single-chain antibody to folate through a reversible disulfide bond. We made this in 2 flavors, and in one case, we also incorporated an endosome disruptor peptide, but we found really that these 2 were equivalent, and the simpler one was easier to make. We confirmed the conjugation in several ways, by spectroscopy, by gel electrophoresis, and we showed that there was minimal or no interference with the specificity of epitope recognition. And here's the thing, it works. So this is the key piece of radiobiological data. This is a clonogenic survival curve in vitro with cancer cells. We've done it with several lines. I'm showing just one. A typical survival curve with a shoulder in the low dose range and then increasing sensitivity as you increase. And clinical radiotherapy is done through multiple fractions of radiation right here in the shoulder range. And so the goal of a clinical radiosensitizer would be to eliminate the shoulder in the cancer cells. And this treatment does. If you compare control treated with experimentally treated, there are ways to quantify this. You take the integral under these curves from 0 to infinity. You come up with a sort of a figure of merit for the treatment. And this is 1.92. Well, you can think of it as a reduction in the dose needed to get a given effect. And that's considered a good figure for a lead compound. It would be something you definitely want to pursue. We know a little bit about the mechanism. We know it does block one of these autophosphorylation events. This is a phosphoepitope that's seen in the DNA-dependent protein kinase by immunostaining in individual cells, and in the treated cells, that phosphoepitope is not detected. So where does this stand? The radiobiology in vitro looks promising. There are various issues with engineering the antibody fragment, with its stability, with modifying it, how you deliver it. That's all underway. I'd like to be able to show you live cell imaging of the uptake and fate, as well as animal experiments, and those are in progress. We hope to have those complete within the next year. And importantly, we've established a discovery paradigm that I think can be extended to other examples of things that one might want to do. We picked a disease, which was cancer. We had a device, which is a single-chain antibody that was modified through the attachment of a nuclear localization sequence and folate and some other things, and we had a delivery method. And in principle, all 3 of these are independent, and you could choose another disease, another device, another delivery method. So I want to speak very briefly about this idea of re-engineering for new function. And I want to tell you that nature's done this with this machine, with the one we're working with. There is a natural example of this having been re-engineered. So DNA repair is, of course, nature's only way to alter gene sequences. And non-homologous end joining turns out to be one of these very ancient functions that you find in many organisms in different domains of life. This Q protein has very ancient homologs. So this was around, right? This problem of double-strand break repair has existed since the beginning of biology, or since the beginning of the DNA world, whenever that came in the progress of biology. And then about 400 million years ago, something happened. Probably a transposable element got integrated into the common ancestor of the jawed vertebrates. And that transposable element encodes a protein complex, 2 polypeptides, recombination activator genes 1 and 2. And all these do is incise the precursor genes of antigen receptors. And those precursor genes already existed in organisms like the lamprey, but they couldn't rearrange to form new receptors, to recognize things that you'd never seen before. And all these proteins do is they make a cut, and then they retain the ends, and they shepherd them into a repair pathway that was pre-existing.